Welcome to this episode of Eureka, which is a COVID special. World and India is facing this huge challenge of novel coronavirus pandemic. World over, the science and scientific institution is trying to find ways of tiding over this pandemic. Of course, the frontline workers are people who are giving medical care, people who are engaged in ensuring hygiene, and administrators, but behind the scene, science is playing a very key and important role. Today, we are going to have this conversation with Dr. Shekhar Mande. He is the Director General of CSIR, a major institution for science and technology in India. Sir, thank you, sir, for being in the show. It's a very wonderful opportunity. And let me start with one interesting question. I mean, your institution is actually an umbrella of organization, lots of organization, more than 40 labs are part of your system. So the very first thing that we came to know is that you said that if you have to fight this pandemic, all these institutions somehow or other have to team up together. So what did you actually do? Why did you thought that it is important that institutions which were working on their own had to come up and then get together?
So what happens is that a calamity like this has rarely been witnessed in the history of mankind.
Yes.
And if you have to fight this calamity, there are so many unknowns. We don't understand much about what it is actually about, what are the issues that we need to tackle, and many different aspects. And if you just leave it to one particular set of people to fight the calamity, it will not be fair. I mean, just to give an analogous example, there's a famous statement that Mark Twain once said that economics is too important a subject to be left to economists.
Okay.
No, I mean, something like that. This is such a calamity, it's too important to be left to only one set of people, one group of people. And therefore, it requires a holistic solution. Okay. And therefore, we decided, and since we span so many different disciplines within CSIR, the 38 labs, we thought that all of us, if we put our heads together, ideas could emerge from anywhere. And to tell you the truth, in retrospect, that was such a fantastic decision because indeed ideas have emerged from some of the most unexpected sources.
Really, sir. I mean, that's very wonderful to hear. And you sort of created something like 5 verticals, right? I mean, so that 5 important challenges, science challenges, this institutions will address together. What are those 5 verticals?
So very early on, even before the WHO was claiming this to be a global pandemic, at that time we had our discussions going on internally in CSIR because we are clearly seeing the writing on the wall. And we decided that to tackle the disease, we should have 5 different verticals. The first vertical is surveillance, including digital and molecular methods.
Okay.
In which we go to fields and then collect samples and then at a very large scale do the surveillance and also do the genome sequencing so that we know how the virus is spreading from which place to another place.
Okay.
The second vertical is on diagnostics. We knew that diagnostics is going to be the key to the issue, and in fact the WHO General Secretary also went on board by then saying that test was the mantra. And we thought that diagnostics is going to be the key, and we must have diagnostic kits, multiple diagnostic kits, which are safe, which are fast, which are cheap, which are accurate. So all those things we should evolve, and we can do that. The third vertical that we decided to work on was interventions. Okay. And interventions principally include 2 kinds of interventions, the drugs, the vaccines. And within the drugs, there are the 4 different kinds that we'll talk a little later. But there is the third vertical. The fourth vertical that we could clearly see that if it were going to be a pandemic, then we are going to have difficulties accommodating so many people in the hospitals.
Okay.
And therefore, we have to have solutions to assist the hospitals. And therefore, we decided to work on hospital assistive equipments, which included protective clothing, gloves, masks, shields, ventilators, and whatnot, oxygen enrichment units, and all those. And the fifth vertical that we could very clearly foresee that anything that you name today, there's going to be a shortfall. Anything. You name a thing, there's going to be a shortfall, which actually turned out to be true a month later.
Yeah.
About a month and a half later, when we look back, that actually is the real scenario. And therefore, we decided that we'll build in a supply chain module for each of the things in every corner of the country. And that was actually very, very wide. Unfortunately, the last aspect that we could not foresee, and we could have built in a 6 vertical then, was an unexpected fallout that scientists like us could not see, was about the reverse migration of people from cities to villages. That also requires technology-led solutions. To support the people who are going back to villages, that does indeed demand technology-led solutions for them. And we could have foreseen then, but we have started work in that area as well. We don't call it a 6 vertical, but we are starting an independent area to work for people in rural areas.
But because it's a very emerging challenge that is coming up, we need to. So what are the lower hanging fruits that CSIR institutions all over the country that provided many states were talking about is that testing. I mean, there was a huge challenge when one had to use a PCR test, that you can't use a regular laboratory for those testing. So CSIR institutions came forward and then they took up on this challenge. Why institution like, a premier institution like CSIR institutions is required for testing? Can't they be done in usual laboratories? That's a question that most people have in their mind.
I'll just quickly tell you about low-hanging fruits and I'll come to testing, what you mentioned. The thing is, low-hanging fruit really are the sanitizers and things like that. And even then we ran out of sanitizers and other equipment. Sanitizer is simple, a mixture of alcohol with glycerine or something like that. But even that we ran out. Now in remote areas, hospitals, police people, army people, they don't have sanitizers. But our labs, because we have presence in places like Jorhat, Palampur, Jammu and all, our labs stood up. In fact, even cities like Lucknow, our labs made several lakh liters of sanitizers and distributed to police force, distributed to army hospitals, distributed to a whole lot of people. So that was a real low-hanging fruit that we could do, but bring in a lot of value. You don't see a deep science and technology in this, but in terms of the social impact is huge. Regarding testing, what happens is that the only gold standard which is available in the field today is what is called as the RT-PCR based test. And it's such an involved test that only the very well-entrenched biology lab can do it very easily. So we decided that because we have some premier biology labs with us, Center for Cellular and Molecular Biology in Hyderabad, or Institute of Genomics and Integrative Biology in Delhi, or Institute of Microbial Technology in Chandigarh, they can be put in action straight away. And it so happened that as soon as ICMR decided that NIV will do testing, we also declared that CCMB will do testing, and NIV and CCMB started to work together almost at the same time. So we are working shoulder to shoulder with other agencies to make sure that the testing comes. But the real challenge also is that we have to train more people in doing RT-PCR tests. Not too many people know how to do RT-PCR and what are the controls and all. So CCMB has trained a large number of people in Telangana on testing itself. So that actually also was, you may call low-hanging fruit, but in terms of social impact, is a huge impact that it creates. It did.
And in times of pandemic, they are the key important aspects, immediate important aspect that one needs to address. Maybe there may not be deep science, but they are immediately needed. Sir, let's talk about the drugs. You said that we'll talk about it a bit later. We know that quite a lot of CSIR Institute is involved in repurposing old drugs to find out whether they can be of use to treating COVID. So what is the lay of the land in regards of drug discovery or repurposing that CSIR institutes are involved in?
So as you rightly pointed out, that CSIR has been involved in the drug discovery since ages. We are one of the first institutions that was involved in drug research and all those things. Many people don't know that we have had very, very famous people who have worked in CSIR who pioneered drug discovery in India. For example, I wonder if you knew that Stephen Hawking. You remember Stephen Hawking?
Yeah.
His father worked in CDRI Lucknow for a short while.
That's a really nice, interesting information, actually.
Sir Ramnath Chopra founded the person who wrote the Drug and Cosmetic Act of India. He was the founder of our IIIM Jammu. Janki Ammal worked in CSIR IIIM Jammu. So we have had actually a great tradition of drug discovery within CSIR in India. And today we also we are the powerhouse of all the drug discovery in India. And therefore we decided that we should jump into this area straight away. We have 4-pronged strategy in drug design. Number one, the new chemical entities, the novel molecule that would fight with COVID-19. Number two is repurposing, because repurposing is actually a very powerful strategy if you want to get something into the market very quickly, and can also be extremely useful. Number three is phytopharmaceutical. Phytopharmaceuticals are extracts from plants, and they can be used, and FDA recognizes phytopharmaceutical as a category. Both Indian DCGI as well as US FDA, they do recognize botanical formulations as drug formulations. And the fourth one is Ayush. We recognize that we have very rich tradition, and whether we can tap some parts of Ayush in this. So they are the four things that we started. But in terms of repurposing, we actually very quickly drew up our priorities. What is going to be the priority of repurposing? And we saw that there was an opportunity in drugs like favipiravir, drugs like arbidol, and all, because they are already in use against influenza. And we started our work very early on those. We developed the processes. We transferred them to the companies, some of the major companies in India. For example, favipiravir, we are working with Cipla very closely together. And Cipla has now started manufacturing that. And we are just starting the clinical trials for this. If the clinical trials results are good, with the best wishes from all the citizens of India, who knows, a few weeks from now, we would have one of the therapies for COVID-19.
So, which essentially means that for this drug, we don't have to depend on any other country, because we have developed our own know-how. We can produce it in India. That's a very key important part of this aspect, right? Though the molecule itself was discovered earlier and has been in use for other diseases, we have the know-how, right?
Absolutely, that's correct. As long as it is off patent. See, if it is under patent protection, then we will not be able to do that. But if it's off patent, of course, we have designed the process for this. So entire favipiravir can be synthesized from all the chemicals that are available within India. Our emphasis from beginning is that whatever we design, whether a diagnostic kit or a repurposed drug or a ventilator, every component has to be available in India. We will not depend upon imports of that. And that has been a strategy of India much before people started talking about how things should be made in India and this and that. So we designed this strategy more than 1.5 months ago, that every component has to be available in India so that we don't depend upon other countries on these aspects.
That's very nice, sir. I mean, we talked about repurposing, but you also talked about, for example, Botanicals. You also talked about Ayush trials, right? So let's talk about Botanicals. I mean, what kind of work is actually going on now?
So Botanicals, there is one particular formulation that we approached DCGI. There is a plant which is found in the tribal belt, all the way from Gujarat, Madhya Pradesh, Jharkhand and all. And it's fairly widely used by the tribals there. We had done the clinical trials on this particular formulation against dengue. There's one major pharmaceutical company in the country, and International Center for Genetic Engineering and Biotechnology, ICGB, Delhi. CSIR IIIM Jammu, and supported by Department of Biotechnology under Ministry of Science and Technology. All of us had worked together on this particular formulation for dengue, and it has cleared phase 2 trials. It's safe for consumption, and therefore we thought because it's an antiviral, works against dengue, we should very quickly try it out against COVID-19 as well.
Okay, that's interesting. So
pardon me?
Sorry, that's an interesting example of how a botanical is, again, for example, maybe developed for some other disease, because we don't even know that such a virus would attack us. So we need to find solutions quickly. So coming to Ayush, the plan is to trial some of the drugs which are under the Ayush?
So what we did is we have a Ayush CSIR joint working committee. And they consist of very well-known Ayush practitioners, Vidyas, from the Ministry of Ayush, taken from different sources, people who have rich experience in Ayurveda. And then we have people from CSIR who actually focus on modern drug discovery and all, who understand the nuances of drug discovery, modern drug discovery. And together, we came up with 4 priority formulations that we should try. And we're actually going to work on that. So one is Ashwagandha, one is Yashti Madhu, one is Guduchi Pipili, and one is a formulation called Ayush 64. And then we designed clinical study from the modern science perspective, exactly the way modern medicines will be tested in the trials, clinical trials. And we identified hospitals And then we took permission for conducting these trials. So these trials should start anytime now.
Okay, so we are awaiting the clinical trial results, and then perhaps we will be able to get even a Ayush drug which can cure at least some aspect of COVID disease. That's a very nice, very interesting.
Fingers crossed, if the results are good, we will get that particular formulation.
Yes, sir. Thank you. I mean, I think we will take a very short break here and then continue this conversation. Keep watching Eureka. We'll take a very short break. After the break, we'll continue and then we'll discuss about how CSIR is trying to develop vaccine, which is, I think, a very key aspect if you want to stop this pandemic. A short break.
भीड़भाड़ वाले इलाकों में ना जाएं। धार्मिक, सांस्कृतिक, मनोरंजन, शैक्षणिक आयोजन से लेकर खेलकूद की गतिविधियों से दूर रहें। संभव हो तो वर्क एट होम करें। काम के दौरान सहकर्मियों से 1 मीटर की दूरी बनाएं और गैर जरूरी यात्राओं से बचें।
Welcome back to Eureka. We are having a very wonderful, very enlightening, very informative conversation with Dr. C Mande, who is the Director General of CSIR. Sir, before the conversation, we were talking about how CSIR is trying to bring in the science and technology, indigenous science and technology power to help the nation face this pandemic in multiple ways. One of the things that is everybody's mind is, when will a vaccine come? What do you think about it? What's the lay of the land of vaccine?
So vaccines, as much as drug discovery, is a very, very challenging aspect. As you would know that I don't want to discourage people thinking that it's not possible. It is definitely possible. The number of vaccines people have designed over the last several decades. But despite the best brains working on things like HIV or dengue, we still don't have vaccines against those diseases. So it's an extraordinarily challenging human endeavor. But nonetheless, we must try our best in trying to get a COVID-19 vaccine. And number of vaccine candidates are going around the world right now. Indian industry is very deeply involved in some of those trials, which is a very heartening thing to see that how Indian industry is actually trying those. In CSIR, we are a three-pronged approach on vaccines. One of them, essentially CSIR is only helping, but it's a major effort by NIV, which is an ICMR institute. It's funded by ICMR. And Bharat Biotech, they are trying inactivated virus particles. But some of that also will be validated within CCMB, Hyderabad. CCMB is our institute, CSIR institute. And CCMB has been culturing the virus in human cell lines. There's something called a Vero6 cell line. It's actually African monkey kidney cell line in which virus can be cultured. And they culture that and then inactivate the virus, and then Bharat Biotech would take it for trying to do the vaccine trials. The second aspect is that once again we are working with Bharat Biotech on what are called as monoclonal antibodies. This has been funded to NCCS Pune and IIT Indore. They will try to generate monoclonal antibodies from recovered patients and see whether these are effective vaccines or not. And the third thing that what we are working on is MW, which actually is difficult to call it as a vaccine, because vaccines are given to healthy people in the hope that they will not contact the disease. But MW is a kind of a formulation that is also given to people who are down with symptoms and things like that. And that clinical trial has already begun for critically ill patients. And the clinical trials should be over another couple of weeks. And we are very eagerly awaiting the results of clinical trials.
So this MW vaccine that you are talking about was actually originally developed for leprosy, right? By Indian effort.
Correct. So it's a bacterium that was discovered in India. The actual correct biological name is Mycobacterium indicus pranae. You know, there's a biological name. And Cadila Pharmaceutical several years ago sourced it from National Institute of Immunology, which is a DBT autonomous institute, DBT funded autonomous institute. And they tried it to generate a vaccine or drug, whatever you call, against sepsis. And the clinical results are very good. So Cadila markets it as Sepsivac, is against gram-negative sepsis. And they have found that the incidence of death rate in sepsis comes down quite dramatically with Sepsivac. Because some of the clinical outcomes of sepsis are similar to COVID-19, we thought that we should give a try to COVID-19. So once again, Cadila Pharmaceuticals and CSIR together have initiated these clinical trials, and they are undergoing multicentric trials right now in PGI Chandigarh, AIIMS Delhi and AIIMS Bhopal. And hopefully within 2 weeks, we'll get the results. And all of us are very, very eagerly awaiting the outcome of these results.
Because one of the major cause of death in COVID cases, I mean, when it becomes critical is sepsis, right? So it's one of the major reason for death. It's not actually the virus, but the overreaction of our immune, which leads to tissue death, which leads to sepsis, right? So that is a huge challenge in treatment, right?
It's called cytokine storm in the technical term. And because of the cytokine storm, the capacity of lungs to absorb oxygen and all goes down.
Okay. So if this clinical trial is successful, then it means that we will be able to recover even more critically ill patient. I mean, the death rate can be substantially reduced. That's what we are hoping.
That's what we are hoping. All of us are hoping, and we need prayers from all of you for this.
Yes, sir. Of course. I think we do see a bright spot in our Indian science. Of course, the medical personnel of India and also the administration is trying their best, which we can very clearly see in the ground. And I think behind the scene, the science has to give the push it requires to face this challenge. You are also talking about the monoclonal antibodies. So these antibodies are produced whenever there is an infection which are particular to that infection. So what you are saying is that, I mean, people were talking about plasma therapy, which is basically transferring blood product, let's say the plasma from a recovered patient to another patient. But you are saying that you are looking at how you can culture it outside the human system. Is that so?
That's right. That's right. So basically, plasma has number of antibodies floating in that. Right? Every B cell produces one kind of an antibody. What we call as polyclonal, because they come from different cells. What we would like to actually identify are antibodies which specifically neutralize the virus. Right? So amongst all these polyclonal antibodies that are circulating in plasma, we only pick up those which are neutralizing the virus, take them out, and then generate that monoclonal antibody and give it to the people who are affected.
So which means that, for example, if we administer it to medical personnel, the chances that they will contract COVID will substantially reduce. So people who are treating can be also saved, right? I mean, that's one important outcome of this monoclonal antibody if we can develop, right?
We hope so. Yeah.
So that's I think a very interesting aspect. I mean, these are all new ways of ensuring that the death rate can come down. Let me now move to another question, which is testing. One of the institute under your system has developed this test called Feluda. And quite a lot of people are hoping that that's going to be a game changer. Can you tell us about what's its uniqueness?
So it's very interesting. We have very bright young scientists across CSIR, of course, but in the institute called Institute of Genomics and Integrative Biology, that's in Delhi. There's a CSIR institute, and two scientists there, Dr. Souvik Maiti and Dr. Debajyoti Chakraborty. What they did, actually, they came up with this fantastic idea that if we put RNA on a piece of paper, which actually has a complementary sequence that is the virus, so that the virus RNA can hybridize with this particular RNA and have a method which can detect that. We could have a paper-based dip test kind of thing. Paper, you dip it in a solution and tells you whether there is the virus or not. It's something similar to the pregnancy test. And they have come up with this test very nicely. It requires CRISPR-Cas technology. The Cas enzyme that they use is their proprietary Cas enzyme that they have discovered themselves.
Okay.
So there's something called Cas9. And that's a very interesting development. We are working very closely once again with the companies on this. And in this case, Tata Sons, they came forward to take the technology from us. And they would be scaling up the thing in the field. As you said, if it's successful, it will really be a game changer in terms of the speed of testing, in terms of the accuracy, and in terms of the cost on all three fronts.
Okay, so today we have this problem of PCR test will detect early, but it will take a lot of time. But whereas the rapid test will detect fast, but you can detect it only after a few days or maybe a week after infection. But this will take away all the, take all the best of both, but keep away, do it fast. That's what you're saying. And that's a combination.
So the major difference between the PCR test and the rapid test that you're referring to are the PCR test try to find out the RNA in the circulating wherever the swab is taken from.
Okay.
While the rapid test that you're talking of is antibody test that actually tries to see whether your body has developed antibodies against the virus or not.
Yeah.
There's a gap between the two. When the virus infects a human being, there is a gap of few days before the antibodies actually develop. IgG antibodies they develop, and therefore the rapid test has its own value, but after a few days of infection. The Feluda that we are talking of also detects the RNA, and therefore it is as quick as RT-PCR. So on the first day you are infected with the virus, you should be able to detect the RNA from the.
Yeah, that's interesting, and because it's a paper test, it can be done very widely. Unlike a PCR test where you have to go for a BSL lab 2, we don't need that. It can be done very fast. That's I think will certainly be a game changer once it comes into the market. After we test and all that. Now let me move to another question. We don't have much time. If you look at PubMed, which is the repository of publications related to biology in a way, a very simple search of novel coronavirus yields you about 11,000 papers just in this year. That is from January to now. That is all over the world. People are working crazy. Basic science. So CSIR, of course, is majorly focused on applied sciences, but there are also a lot of institutes which just does world-class basic science. So where do you see CSIR in this game of understanding the virus, understanding the complex biology of the virus, and then being ready for, let's say, surprises?
So basically, as you said, a lot of our emphasis has been because of the scale or the magnitude of the disease has been on translational aspects right now. But notwithstanding that fact, many of our people are also working on fundamental aspects of understanding the virus. Many of our people have started sequencing the virus. Today, we have submitted about 103 sequences in the GISAID database, the viral sequences, but 100 more are ready to be submitted. So we have about 200 sequences. And there is a group which is frantically working on how our sequences evolving and this and that. There are also people who are working on other different kinds of aspects of the virus, fundamental aspects of the virus. And those papers are already in the pipeline. We must realize that some of the countries had a headway because China, it was detected more than a month before what we detected in India. So they had a headway in starting the basic also work on that, and many other countries. So, but although we were there right up there at the front when everything began, our scientists are working very hard on even the fundamental aspects of the virus, understanding fundamental aspects of the virus.
That's very nice, sir. That's nice, sir. Last question. This is more for our audience. A lot of people have fear. Fear is the first thing that people comes to a mind when we talk about this pandemic. But as a scientist, as a person who is heading a very key scientific department of this country, what do you suggest to us?
I think that fear is partially unfounded and partially true. Unfounded because people should recognize the fact that 80% of those who contact the virus will not even show any serious symptom to be hospitalized.
Okay.
So 80% of the population who contact the virus can happily recover sitting at their home itself.
Yeah.
So there is nothing to worry about that particular aspect. You will have some symptoms. You may have some cough and cold. You may have some loose motion. You may have some slight fever, but you will recover on your own. That's 80% probability, which is very good. Less than 10% people may have to go to hospitals, and even much less eventually would go to ICUs and would eventually succumb to the disease. What we should make sure is that we don't contact people. There's a certain thing called high-risk population. We definitely keep them away. So the comorbidities are known, for example, with diabetes and hypertensive people. So we should keep them away from contacting the disease because they are a higher probability of doing that. But for most people, absolutely nothing to worry because you will only develop very mild symptoms and you will be cured of that. So in that sense, there is no reason to panic. At the same time, there is a reason to be careful to make sure that people who are at high risk, you keep them away from contacting the disease. And only powerful way of doing that is social isolation, what we call, and social distancing norms. If we do that, isolation is not a correct word. Social distancing, if we follow religiously, I think we will win this war comfortably.
Thank you, sir. Thank you for this opportunity. Thank you for your time. It's a very wonderful conversation. We came to understand quite a lot what this Indian science, particularly the institutions under the CSIR umbrella, is doing to face this pandemic. Thank you for being in the show. Keep watching Eureka. We'll come back with another interesting conversation next time, same week. Thank you, sir.
Yeah, thank you.